DFG Science TV, Function Through Diversity, pioneering work in China, the largest ever forest experiment on biodiversity.
In the spring of 2009, the international research team started a unique forest experiment in eastern China. On an area covering over 50 hectares, they launched an experiment to investigate the role of biodiversity for ecosystem functioning of forests. They want to find out whether the functions of different species in an ecosystem complement each other, and in what way complementarity might be achieved. Does an increased number of species help the ecosystem to function better? To find an answer to this question, they need to know the traits of the various species. No easy task, given that very little is currently known about the species in the forests in the east of China. This means that the researchers need to examine the 150 or so species of trees and shrubs that occur in the natural forests to record a whole raft of characteristics.
So now I try to get at least 10 of the leaves for later analysis in Halle. And I try to get 10 developed ones, just like this one here. I don't try to get these, where you can see some signs of herbivory, or leaves like this, which are not fully developed.
The term trait can be defined as a measurable biological property. All of the traits taken together give the plant's phenotype. How diverse the species here can be in terms of their traits can be well demonstrated by these differently shaped leaves. As well as the shape of the leaves, the researchers also examine a lot of other characteristics, such as the specific leaf area, for example. This is calculated using a simple formula. The leaf surface area, divided by the dry weight of the leaf, is a measure of the area of foliage for collecting energy that a tree with a certain amount of biomass is able to produce. In the natural habitat of the trees and shrubs, the researchers also determine the rate at which the tree's branches grow and how many leaves are formed each year.
We are in CSP8 again, or still, and what I'm doing right now is to measure the twigs, which I labeled last year, what you can see here. And I'm measuring the length of the twigs, and I count the number of the leaves. So in this case we have 1, 2, 3, 4, 5 leaves. And the length is 6.5 centimeters.
The species also differ in terms of their wood, for example in wood density or in size and layout of the water vessels. In addition, the researchers determine the age of the trees by counting their annual growth rings. Most of the work is done back in the lab. There, the researchers prepare the leaf samples so that they can analyze the nutrients in the leaves, for instance. They also conduct experiments on many of the tree and shrub species in the greenhouse. Here they can grow them under controlled and different environmental conditions. This enables the team of researchers to see how each species adapts its characteristics to the prevailing environment. Under the different conditions, the plants have different specific leaf areas and different growth rates. The researchers need to know all of these traits so that they can calculate the level of diversity of traits represented by all of the species in a plot. This so-called functional diversity is probably more important for ecosystem functioning than the actual number of plant species. It may be, for example, that two species in a forest have such similar characteristics that they perform the same function in the ecosystem. In that case, one species could replace the other. This is another reason why it's so important for the researchers to analyze the plant's wealth of traits. The results of these experiments thus make a significant contribution towards the overall goal of the project as a whole, to discover the impact of tree and shrub diversity on ecosystem functions in one of the most important biodiversity hotspots on Earth.
Visit DFG Science TV for more information. Awaken the researcher within you.